Science is really the foundation of anti-doping. We need to be able to detect and deter literally hundreds of substances that an athlete may be using to dope or manipulate their performance.
One of the keys to that detection? Laboratories like the one housed at the Brian S. Finkel Building in Salt Lake City, Utah. Inside, you'll find the Sports Medicine Research and Testing Laboratory, routinely referred to in the anti-doping field as SMERTL. SMERTL is one of the premier anti-doping labs in the world and just one of the Partnership for Clean Competition's many research collaborators around the globe.
The Partnership for Clean Competition is really on the cutting edge of technology and funding cutting edge research and science that is able to work within the anti-doping space.
The focus is moving the sports drug testing field forward as a whole, working together with funding partners and talented researchers alike looking for new solutions.
There's been a lot of really good improvements in the last 20 years in anti-doping science and techniques. But, I think some of the things that we're most proud of is the alternative matrices. And so these are looking at non-traditional testing matrices. So most of the testing was usually done in urine and then in also blood that's drawn from the arm. And so there's been a lot of advances in looking at alternative matrices. And the advantage of these things are they're less invasive for some of the testing programs. So the athletes, it's easy for them to test. You can do the testing anywhere in the world. They're cheaper and more convenient to do some of these things. And importantly, some of these alternative matrices will also give you different information. So you can then hone in on different periods of time on use and so forth. And so I think there's been some great improvements in that area that we're very proud of.
One of the things that we're most excited about is changing the experience that athletes have around blood collection. And specifically, this includes new ways of collecting blood called dried blood spots. This is a way of collecting blood rather than sticking a needle in an athlete's arm. We can collect a small amount of blood through a device that either pricks the finger or actually attaches to the skin on the athlete's arm. We can collect about 20 times less blood than we're collecting now and analyze that for prohibited substances and be able to effectively detect those substances in a much smaller volume.
Overall, the athletes are super positive about DBS collections. They're not invasive, they're virtually painless, and this means that there's really minimal impact to training, to their daily life.
DBS collections have improved my experience as an athlete because they are super easy, they are efficient, and completely painless.
Of course, it takes a lot of dedicated time, hard work, and expertise to go from a concept to a fully validated, ready for primetime anti-doping solution. An amazing example of the translational research the PCC is proud to have supported from the beginning.
When we had first started, there were ideas of certain drugs that we might be able to detect in dried blood spots, and certainly over the last 7 or 8 years, this work has become a lot more in depth, where laboratories have been able to validate assays for different detection methods, different drugs. So where we've started here specifically is a lot with the growth hormone assays that we're looking to detect in dried blood spots.
DBS in an anti-doping context has evolved immensely in the last 3 to 4 years. We've done a lot of research that's applied specifically to detection, developing new detection techniques that deal with those smaller volumes of blood.
Great science takes time. Therefore, PCC-funded scientists at SMERTL and around the world have worked tirelessly to validate DBS as a new testing methodology. The PCC partners have a vision of replacing painful and invasive venous blood collections with a focus on dramatically improving the athlete drug testing experience.
Interestingly, the success of DBS also couldn't be realized without collaborators in the industry. Therefore, the anti-doping community has worked closely with a startup company called Tasso, who developed a reliable and safe DBS collection device for clinical medicine applications. The Tasso collection device sticks directly to the skin on the upper arm, and with the literal push of a button, painlessly pierces the surface of the skin to collect a micro volume of capillary blood for testing, capturing the blood on multiple absorbent pellets in a sealed cartridge. In less than 2 minutes, the full DBS cartridge is transferred into a tamper-evident DBS security kit from another private partner, a company called Innovero. The kit is then sent off to the lab. With experience, researchers have established an efficient DBS workflow to open the secure DBS kits and process samples through the lab for analysis.
It's not much different than a urine sample. So the samples will come in just as normal through some kind of shipping organization. We'll log those samples into our system. what we call accessioning. We'll extract those samples or aliquot them out so that each test that needs to be performed on a spot can be done. We'll run the test that's required, and then we'll get the results and report them out.
Getting DBS to this point required multiple projects, including a massive PCC-coordinated field study with Major League Baseball. But after thousands of athlete samples collected and tested, DBS has been validated as a robust and athlete-friendly collection method.
It was shocking to me what a big deal this was.
Having worked with the PCC all the way through the development of the DBS collection and analytical methods, professional leagues like Major League Baseball, as well as the Olympic and Paralympic movement, were poised to spearhead their use.
We are using dried blood spot technology across all levels of our anti-doping programs. At baseball, we have a number of programs in place covering prospect players that are coming into our industry all the way up to our major league players. And as of the most recent collective bargaining negotiations that have been completed, we now have dried blood spots in place from the lowest level all the way up to the highest.
It was really the pandemic that gave us the kickstart to really get DBS out into the field. We decided to go big with our DBS collections. We had 1,000 samples in our test distribution plan. This gave us a lot of exposure to DBS for athletes in the field, and also provided a lot of samples to really drive the research and the analysis methodology development in the labs so that they could really bring DBS up to the forefront and make it a usable tool in the future.
You know, we're not going to move forward with anything until our expert advisors tell us that it's validated and the science is strong enough where we are not going to lose the ability to detect and to deter growth hormone use. But through many years of investigation in the field, also relying on our scientific advisors at the laboratories, we are finally at a place where the analytical technology is strong enough where we were comfortable making the switch.
And switching to DBS pays dividends. It strengthens testing regimens, testing for performance-enhancing substances such as anabolic agents, hormone and metabolic modulators, and growth hormone, as well as others that are unstable in urine. All of this can be achieved in DBS. This adds value to the anti-doping toolkit and keeps us ahead of the cheaters by increasing analytical capabilities and deterrence efforts.
From a blood testing perspective, we're able to collect more tests more frequently, have an increased deterrent effect because we are collecting more samples per athlete, and we're able to effectively detect some of those hard to detect substances such as growth hormone that can only be detected in blood.
Plus, there's the economics.
We are certainly focused on the science and running the strongest program that we possibly can. We also have to look at the dollars. And intravenous blood draws from collection through shipment, through equipment, all the way through the laboratory process is incredibly expensive. And being able to make that switch to a process that is exponentially more cost effective not only allows us to continue to run the same strength of a program, but we can then reinvest all of those dollars that we're not wasting on intravenous blood draws and put them into other areas of our programs. Considering that you can do so much more testing in exchange for so little resources without having any drop off in deterrence or integrity, it was a very easy decision for us, and I would recommend that other programs make a similar decision on their end.
The PCC prides itself on helping to make that decision a straightforward one that is proven with world-class science. With dozens of scientific publications and presentations supported by the PCC and others, DBS is a translational success story.
We've been on the cutting edge of alternative matrices like dried blood spot technology that has really advanced what we are able to do in the anti-doping space.
We wouldn't be where we're at today without the PCC's continued support. And so we've had funding back from the PCC at least for 11 years since I've been at Smyrtle, and we've done many different projects regarding dried blood spots, from blood doping projects all the way through to growth hormone detection programs. And so without the PCC's continued support, we wouldn't be nearly where we're at right now.
The validation process for a new test or a new method takes a very long time. And with the PCC's support on the financial end, but also just the logistical end, we have been able to get the dried blood spot collection and analytical method validated in a really impressive time frame, very compressed time frame, to the point where, as opposed to a decade or longer for the validation of certain methods, we're looking at just a couple of years where we were able to get to a place that we were comfortable adopting it.
All of this, all of the progress, serves as the PCC's blueprint for the next major leap forward in anti-doping.
We want athletes to be confident that when they step onto the field of play, we've got an anti-doping system in place that levels that playing field through effective science, both detection as well as deterrence.
With being able to test more blood and having it being tested in this super easy, quick, and efficient method, it will just 100% help level the playing field, because we'll be able to get a lot more blood tests done.
That is the promise of clean competition.